name is Kristoffer Gonzales from grade 11 St. Theodore. Lesson 13.2. The role of antibodies in the immune system. Our topic for today is the type of immune responses in plants and animals. The role of antibodies in the immune system. The role of T cell in the immune system. Vaccine works on the concept of antibodies, which are proteins that the immune system produces to help defend the body. Vaccine works by using an inactive virus so that our body will produce an antibody to fight the incoming active virus. In light of COVID-19 pandemic, learning more about how antibodies protect the body is even more important. If we know the importance of the vaccine and how it is used, we'll not be afraid to get the vaccine like COVID-19 vaccines. Components of adaptive immune system. First, antibody-mediated immune response. Second, cell-mediated immune responses. B cells. B cells, which are type of lymphocytes mature in the bone marrow. They are the ones that are responsible for the secreting antibodies, which are also known as immunoglobulins, in response to pathogenic invasion. B cells are type of lymphocytes that are responsible for the humoral immunity components of the body, adaptive immune system. These white blood cells produce antibodies, which plays a key important part. Hematopoiesis. White blood cells that starts off as a hematopoietic stem cell, which develops into either myeloid progenitor or lymphoid progenitor. The differentiation of these cells is a process called hematopoiesis. Hematopoiesis, the formation of blood cellular components, occurs during embryonic development throughout the adulthood to produce and replenish the blood system. These are the products of hematopoiesis. These are processed in the antibody-mediated response. B cell activation, plasma cell differentiation, and clonal selection. B cell activation. Naive B cells are activated for differentiation when they bind to pathogens. B cells are activated when their B cell receptor or BCR bind to either soluble or membrane-bound antigen. Plasma cell differentiation. Active B cell can differentiate into plasma cell that produce large amount of antibodies. The differentiation of B cell to plasma cell is an essential process for humoral immunity. The process of clonal selection. The process of presenting the antigen and selecting the cell that can respond to this antigen. A clonal selection hypothesis may explain why secondary immune responses are so effective at preventing reinfection by the same pathogen. Antibodies. Antibodies are Y-shaped proteins that B cells release in order to bind to specific antigens. Antibodies are created in response to the presence of antigens, but these do not directly destroy the pathogen. Instead, they, they serve several functions that help other cells and process in destroying the pathogen. An antibody is a protein component of the immune system that circulates in the blood, recognizes foreign substances like bacteria and viruses and neutralizes them. That's all for my report. Thank you.
Good morning, sir and classmates. I'm Aliaza Marcelino and let's learn with me. In this lesson, we're gonna tackle about the features of antibodies, structure of antibodies, and function of antibodies. So, next page. The features of an antibody. So, as you can see, we have four features of antibodies: the specificity, memory, self-tolerance, and structure. First, what is specificity? Specificity is an antibodies bind to the antigens that they were specifically created to target. So, nangangahulugan na ang specificity ay degree na kung saan ang immune response ay nagdi-discriminates between antigenic variants because antibody specificity can either be viewed as a measure of the goodness of fit between the antibody combining site or paratope and the corresponding antigenic determinant or epitope, or it is the ability of the antibody to discriminate between similar or even dissimilar antigens. So next page. Let's look at the picture. Antigens are parts of the pathogen that tell your body it's being attacked. Basically, by binding to specific antigens, antibodies eliminate foreign substances. Kapag ang pathogen tulad ng harmful bacterium or virus ay pumasok sa katawan, the variable region of the antibody binds to the pathogen at eventually nagkukos ng variety of actions tulad ng eliminating the pathogen. Dahil kinikilala lamang ng isang antibody ang specific antigen at antibodies that attack cancer cells. At ang halimbawa nito ay do not attack normal cells. Ito ay tinatawag na specificity of an antibody toward that antigen. So next page. Let's look at the picture. In this way, inaalis ng mga antibodies ang mga pathogen na nakakapinsala sa katawan ng tao sa pamamagitan ng particularly recognizing and identifying bacteria and viruses. Also, an antibody specificity ay tinutukoy sa pamamagitan ng pagkukumpara ng binding signals in cells expressing the target protein to control cells with the target gene. Dahil a highly specific antibody, dapat hindi magpakita ng binding activity if the target isn't there. So next is memory. Memory B cells can specifically remember the presence of any repeated infections. So memory is the ability of the immune system to respond more rapidly and effectively to pathogens that have been encountered previously. Also, memory responses, which are called secondary, tertiary, and so on, depende sa number of exposures to antigen. This is particularly clear in the case of an antibody response na kung saan ang mga characteristic of antibodies produced in secondary and subsequent responses ay naiiba from those produced in the primary response to the same antigen. So next page. In this page, the immune system can remember previously encountered pathogens. And memory B and T cells are critical in secondary responses to infection. So, isa sa mga hallmarks of our immune system ay ang kakayahang makaremember ng past exposure to pathogens. Tulad ng exposure ay maaaring mula sa infection or vaccination. And by remembering, we are ideally fully protected from infection upon future encounter with the same pathogen. Although Humoral immunological memory is mediated in part by serum antibodies secreted by long-lived plasma cells or LLPCs. As you can see sa second sentence in our PPT, na ang mga cell na ito ay usually not described as memory B cells. Instead, the memory B cells are defined as long-lived and quiescent cells that are poised to quickly respond to antigen upon recall. So next page is self-tolerance. What is self-tolerance? Self-tolerance antibodies only usually target foreign bodies and substances in the body. So self-tolerance ay ang ability ng immune system to recognize self-produced antigens as a non-threat while appropriately mounting a response to foreign substances. This balance of immunological defense and self-tolerance is critical to normal psychological function and overall health. Basically, self-tolerance mechanisms play roles in cancer, autoimmune diseases, colonization by commensal bacteria, and pregnancy. So, next page. If an antibody to work against the body's own healthy tissues, then this is known as autoimmunity. But what is autoimmunity means? Next page. So, as you can see, autoimmunity, it is a condition in which the body's immune system mistakes its own healthy tissues as foreign and attacks them. Most autoimmune disease cause inflammation that can affect many parts of the body. Kapag meron kang autoimmune disorder, your immune system does not distinguish between healthy tissue and potentially harmful antigens. As a result, the body sets off a reaction that destroys normal tissues. The exact cause of autoimmune disorder is unknown. So here are the structure of antibodies. Antibodies are made up of 2 light chains and 2 heavy chains of polypeptides. Each chain of polypeptides has a constant region and a variable region. So antibodies are immune system related proteins that called immunoglobulins, and also each chain of polypeptides has a constant region and a variable region. So what antibodies known again? Let's see in the next page. What's antibody known as? An antibody is also known as an immunoglobulin, a large Y-shaped protein produced by B cells and used by the immune system to identify and neutralize foreign objects, such as bacteria and viruses. Remember that an antibody, formerly called immunoglobulin, is a large Y-shaped glycoprotein produced by B cells and used by the immune system to identify and neutralize pathogens. Antibodies are produced by B cells and are either secreted into circulation or remain expressed on the surface of the B cell. So next page is, now let's talk about the parts of an antibody. They are antigen binding site, light chain, and heavy chain. So each part of an antibody consists of 4 polypeptides. Again, they have 2 heavy chains and 2 light chains joined to form a Y-shaped molecule. The amino acid sequence in the tips of the Y varies greatly among different antibodies. This variable region composed of 110 to 130 amino acid that give the antibody its specificity for binding antigen. In this page, we have the 5 distinct types of an antibodies. And what are those? Let's find out. Let's see the pictures. So there are 5 types of heavy chain constants, regions in antibodies, and according to these types, they are distributed and function differently in the body. 5 different isotypes of an antibodies, each perform different function and are generally found in the different parts of the body. Human antibodies are classified into 5 isotypes: IgM, IgD, IgG, IgA, and IgE. First is IgG. What is IgG? It is the main antibody in blood. It has a powerful ability to bind to bacteria and toxins, and thus it takes on an important role in the biological defense system. So it is the only isotype that can pass through the placenta, and also it is transferred from the mother's body to protect a newborn. Second is IgM. What is IgM? It is constructed of 5 units of basic Y-shaped structures and is mainly distributed to the blood. So IgM produced first upon pathogen invasion by B cells, and so it has a key role in the initial immune system defense for protecting the body. So third is IgA. It is present in breast milk and protects the gastrointestinal tract of newborns from bacterial and viral infection. So while in blood, IgA is present as monomers. It is the shape of a single Y, but it forms dimers, a combination of 2 Ys. In secretions such as bowel fluid, nasal discharge, and saliva, to prevent bacterial invasion from a mucous membrane. So fourth is IgD. What is IgD? It is present on the surface of B cells and monomeric antibody isotype that is expressed in the plasma membranes of immature B lymphocytes. So it is reported to play a role in the induction of an antibody production and the prevention of respiratory tract infections. So last is IgE. What is IgE? It is believed that it was originally related to immunity reaction to parasite. By binding to mast cells, IgE is believed to be involved in allergies such as pollinosis. In this page, we're gonna recall the summary of 5 distinct types of antibodies. So, for the summary of 5 distinct types of antibodies, remember that IgG is found in your blood and tissue. IgM is mostly found in your blood. IgA is found at highly levels in fluid or mucous membranes, makes such as saliva, tears, and nasal secretions. And lastly, IgE is mostly attacked to immune system cells in your blood. So, let's go to the next page. Agglutination. What is agglutination? As you can see, it is agglutination. It helps protect the body from harmful substances, toxins, and pathogens. This process also forms the antigen-antibody complex. Basically, the antigen and antibodies combine by a process called agglutination. It is the fundamental reaction in the body by which the body is protected from complex foreign molecules, such as pathogens and their chemical toxins. So next is the function of an antibodies. In this page, we're gonna tackle about neutralization, complement fixation, and opsonization. So let's start with the neutralization. Neutralization, antibodies bind to pathogens and block specific sites in order to lower their capacity for infecting the cells of the body. So antibodies contribute to immunity in 3 ways. Preventing pathogens from entering or damaging cells by binding to them, neutralization, because neutralizing antibodies are naturally occurring antibodies that play an important role in the immune system. They work alongside binding antibodies that signal the presence of an pathogen in the body, so that white blood cells can locate and kill it. In the next page, a neutralizing antibody, or NAB, is an antibody that is responsible for defending cells from pathogens, which are organisms that cause disease. They are produced naturally by the body as part of its immune response, and their production is triggered by both infections and vaccination against infections. While neutralizing antibodies can result in lifelong immunity to certain infection and can be used to see if a person has developed immunity to an infection after they have recovered from it. Also, neutralizing antibodies are responsible for blocking the entry of a pathogen into a cell. So, that is firstly unable to infect healthy cells, and secondly, it is unable to replicate and cause severe infection. Complement fixation. It is antibodies activate the complement system of the innate immune system through complement fixation. It triggering destruction of pathogens by stimulating other immune responses such as the complement pathway, because the complement system starts a long cascade of proteins production that either opsonize a pathogen for phagocytosis or lyse it directly by forming a membrane attack complex. As you can see in the picture, the process of binding serum complement to the product formed by the union of an antibody and the antigen for which it is specific that occurs when complement is added to a mixture. Also, complement fixation is used to detect the presence of either specific antibody or specific antigen in a patient's serum based on whether complement fixation occurs. In this page, we're gonna tackle about opsonization. It is the antibodies can coat the surfaces of pathogens, and this process marks these pathogens for destruction through the process of it. Basically, opsonization stimulating removal of pathogens by macrophages and other cells by coating the pathogen. During opsonization, the antibody expresses the tail for an Fc receptor on a macrophage, neutrophil, or natural killer cell. Antibody opsonization is a process by which pathogen is marked for phagocytosis. Let's look at the picture. As you can see in the picture, opsonization is defined as the coating of pathogens with antibodies in order to increase their susceptibility to ingestion by phagocytes. Facilitation of phagocytosis is called opsonization. Opsonization involves antibodies marking the pathogens for destruction by phagocytic cells. Phagocytic enhancement by antibodies is called opsonization. It is true because antibodies also mark pathogens for destruction by phagocytic cells, such as macrophages or neutrophils, because phagocytic cells are highly attracted to macromolecules and antibodies. If may mga hindi kayo maintindihan, you can read here the summary of the lesson. You can read it by yourself. Pero di pa po nagtatapos ang report natin dyan. Because we have activity just to make sure na may natutunan kayo. Just answer this and click the pause button para masagutan mo at nasa end ng video ang tamang sagot. And that's all our report about lesson 13.2, the role of antibodies in the immune system. Thank you for listening and I hope marami kayong natutunan. I'm your reporter Aliaza Marcelino from STEM 11 St. Tudor. So here's the correct answers. Thank you for participating.